Isabella. Branch
Thank
bamboo.
you.
So any bamboos that we see off transect, we should definitely have a look at. I know that the sea
Okay.
pens like that we've been going past, I believe Meredith has lots of samples from the trawl survey, so are not high priority for collections, but any of the bamboos we should definitely take a look.
Okay.
And if there's anything that's seen that is very high priority and we're not sure if we're going to see again, I can drop a target and we can, if it's of interest, we can go back to it.
Sure. That's good too.
That was great. I think, was it you, Sam, who was on when you came back to that stalked farrier, or the branching farrier, sorry.
Yeah. That one
kind
was,
of worked well, backtracking to it.
Yeah, especially since it was so close to the end of the line. Right now we're at 120, so. But
little Gersemia.
Easy to back up.
Have a look if Garcini's on the list.
Is that a black coral we're just going over? Or no, or was that the Gersemia you saw?
There was the Gersemia was pink and translucent.
That's what it was.
See a
Yep.
black coral.
Yeah. Yeah.
I know we sampled Gersemia
It's sort of blurred on my, kind of blurred on my screen, so it resembled a pinkish black coral to me, but that's, I think you have a better view. Does it seem steep? I know it is a slope and it looks steep on the map. How does it seem to you guys navigating this?
Looks moderate. I wouldn't say it's steep. It's pretty moderate.
Okay.
Yeah, we've ascended maybe 10 meters or so, 15, something like that.
It's definitely steeper right here, though. Yeah, we suddenly have come to it.
Oh, yeah.
Looks like you're dragging the camera on the ground.
Yeah, got steep all of a sudden.
There we go. Another thorny head. Another. These little, Abby, did you mention the sea cucumbers with the frilly white stalks? I always am fooled by those sometimes. I have to be reminded that those are sea cucumbers.
Yep. Well, they're unusual because they're attached. Little stars.
Yeah.
And apparently they're quite, they have sort of almost like a shell-like hard textured surface. Not squidgy like most of the other cukes. Thorny heads.
Thorny heads, yep. Is this another Gersemia you're seeing, Steve?
Yeah, I think that looks like it. We sampled one up in Olympic Coast. I don't know if we have any down here.
I believe we do. For those watching, we're in the middle of, well, not quite in the middle of, but we're conducting a quantitative survey transect where we transit the ROV at a steady speed and height above the seafloor as best possible. And those two little dots in the center of the screen that you're seeing are lasers. They're spaced 10 centimeters apart, or about 4 inches, a little less than 4 inches. And they help us size things. So after the cruise, a group of video analysts will review this video and count and estimate sizes of all the critters we're seeing here and classify the habitat. The transect will be about 200 meters in length, and we can calculate the width of the transect from the view. So we get an area, we get an estimate of the area covered, and from that we can calculate densities of all these various critters that we're seeing. And then that makes those density values are comparable with other areas that we've surveyed and done conducted these transects in the same way.
And Di, when you said that we were in the middle of a transect, we actually were right at the middle of the 200-meter transect. So
Oh, we were.
We were. It was perfect. So
Okay.
now we're at about 80.
Well, that was a good guess.
Yeah, 80 to go.
I should minimize and look at, is the map up on channel 3?
Yes.
Yep.
Have a look at that and see.
And you can
what I
see
can,
the radius of 200 meters that's currently up. So our start point was a little triangle, kind of in the middle here of that circle. And we're moving to the northeast right now towards target or waypoint 3B.
Okay.
There's some Swift, yeah.
Oh yeah, good. We're making good timing toward 3B. That's great. We wanted to, have the transect end near there, so we're right on course. Perfect.
Yeah, we'll end the transect right on 3B actually. So turned out well.
Perfect.
It's almost like it was measured that way.
Almost like it.
Well, Lisa, when she planned the dive, knew that it would be around 230 meters, I think was the estimate, as up the straight line up the slope.
Quite interesting.
Coral that we just went over.
Yeah, coral and sponge.
Yeah.
And so we did not collect any Garcinia when we were on San Antonio Bank before, so that would definitely be a potential collection.
Okay. So hopefully we'll continue to see them, and we're getting closer to the end.
It seems to have been fairly consistent, the habitat coming up this slope. That looks like a little predatory sponge there as well.
Mm-hmm.
I can't help but get a laugh out of the predatory sponges. Little sponge thugs.
Yeah. They do, when you look at their spicules under the microscope, they are pretty fearsome. They like properly look like fangs.
Really?
Those little hooks, yeah.
And who do we have swimming right towards us?
So that just went under the lasers there too.
Yes.
The little yellow dots. Well, I thought that they were polymastia on the last dive, but then we had a zoom, and I'm not so sure. So yeah, keep your eyes out for little clusters of yellow dots like that. Be nice to have a zoom on. This white fella on the right as well. I'm not sure what that is.
Okay, I saw those little yellow gumdrops earlier as well.
Yeah. Yeah. Pretty common, whatever they are.
Lisa's still on the chat with us, so she can help weigh in on. Oh, I see an egg case in that sponge off to the left.
Yeah, I know that there was some comment in the dive plan that we could go and explore southeast of 3B as well. little suggested shrimp, a little pink flash going across the screen.
Guy Crofton says his video locked up. Hopefully you've been able to get back on, Guy. Guy is with the U.S. Geological Survey, and he joined us on the science chat and is watching.
It's really useful to have that geological perspective because it really is the underlying geology that kind of dictates what kind of biological communities and assemblages we're going to be seeing.
Yeah.
Whether it's methane venting seep sites, where you've got methane that's venting up through the accumulated sediments, or these rocky ridges, or kind of more cobbly areas like we're seeing now.
Yeah. And there are certain fishes that are found in these various habitats, the rock fishes I'm thinking of in particular that I'm most familiar with. Some are mostly found in very high relief rock. And I noticed it on one of the earlier dives that we did. We went from cobbly, lower relief rock, and there were different rockfish there than there were in the higher relief rock. But they really do have their preferences. That's that lovely hagfish off to the right. Another little thorny head.
Maybe a Dover sole. Didn't get a good look.
Did I miss it? There, I didn't see it.
It was off to the right. I just saw a tail.
Ah.
Predatory sponge, another Thinea sponge. So any collections we do after the transit will also help when the analysts are going back through and recording all the fishes and any invertebrates bigger than 10 centimeters. It will help them make those identifications because they'll have a kind of a specimen to go along with the imagery to be able to say, right, when we were looking at that white sponge there, well, now we know that's what it was, or it was definitely this rather than that. Because often on imagery it can be pretty difficult to know for sure. I know on the AUV sometimes we're sort of struggling even to look at whether something is a sea pen or a sponge. It's really nice to be able to have those samples to confirm.
Yeah. These visual surveys that we're conducting like this have really taken off in the last 10 years, 10, 20 years with the advancement of these technologies. It used to be that all we knew about the deep sea, in particular corals and sponges, was what came up in trawl survey or commercial trawl samples. And of course, those specimens were often, pieces broken off or sort of mangled and could be difficult, but that was what most of our knowledge was based on. And so these underwater visual technologies that have taken off have really been expanding our knowledge at a rapid rate, haven't they, Abby?
Yeah, and I'd also say it's allowed us to increase our knowledge of what species are occurring in these rocky habitats, which are not accessible to trawl surveys too.
Exactly.
I was looking, it was kind of interesting. So a lot of the first sponge collections off the West Coast were done by the research vessel, the Albatross, which I believe was the first vessel that was built specifically for marine research. And one of the fun things you get to do when you're looking at sponges is go back through these old papers and yeah, a lot of the descriptions of some of these West Coast species were collected around 1900 with some of those first albatross surveys off the West Coast here. And then there's kind of a big gap, but now things are really accelerating again because we've been able to take advantage of, vehicles like Hercules and vessels like the Nautilus, which are equipped to go and get those images from the seabed and also pick up specimens. And as you say, dive very delicately rather than just, do a trawl and pick out the pieces.
Yeah. And by the way, Bob, several friends of mine have been watching with great interest, and they're so impressed by how delicately the arm of the Hercules can be maneuvered to collect samples. They're just really wowed by that. So a lot of people have been fascinated with the sampling efforts.
Transect complete.
Yay!
Oh, Bob, you're on mute. So science, do we want to look around here or continue on to our next waypoint?
Okay, so my V-Link is popping in and out. I just got back in. I was out for a few seconds.
Alrighty.
So let's see where we are relative to waypoint 3B.
Sure can. We are at waypoint 3B. Would you like to explore here or continue on to our next waypoint?
Zoom in out.
We had instruction that if we were in really interesting stuff, and Guy Cochran is advising us not to explore around here too much, unless, Abby, there's something here you see that we should collect. I'm looking at the chat right now.
I think just because we are seeing a lot here, and I know that previously sometimes if we move on, we things change. But if we do see there's some predatory sponges, it would be nice to get a sample of one of those from this location. I think the Garcemia, if we see it again. So maybe as we wander towards 3C, but they seem to be pretty common, so I don't think we'll need to go too far. Maybe if we can stop in an area, we might even be able to collect a few things. If there's a cobble with one of those.
Finish the zoom in. Got a little bit of lens left.
Okay.
Is that it? Is that our target?
Let's see, I'm looking at the chat right now. Daniel Geigers asks, are you aware of Valero 3 and 4 sponge samples? I guess that question would be for you, Ashley.
Alrighty, so science, we can start moving along this contour line. That's potentially a little bit of a ridge or a little bit of a slope, upslope. Perfect. I will start moving us that way. We're going to do 325 pilots, bridge nav. 30 meters, bearing 325.
Oh, that's a nice sun.
That's a beaut. Yeah. Nice zoom.
Okay, zoom out.
I'm going to mute and type a question to Lisa for a moment. So.
I sense another zoom coming.
Yeah, you can zoom in again.
Ship move underway. Oh, are we okay to also let the bridge, they can let them know they can change their heading?
Yeah.
Great.
So Sam, if we want to stop and sample, is it just a question of letting you know as soon as possible?
Stand by. Stand by. Sorry, Abby, could you repeat that?
Just once the ship moves on underway, is it just a question of letting you know as soon as possible if we want to have a closer look at something?
Yep, absolutely. Anytime
Yep.
we can stop.
Okay.
So just
Hey,
let us know.
Shore side, there's that asbestopluma.
Yeah,
We're
we're
gonna grab one of those.
Yes, please.
Which is the asbestopluma?
White stick. Where's my telestrator pen?
And this is a sample that we want to collect?
Yep.
So we want to
Yes,
cancel
please.
that ship move? Yeah.
Good eye, Steve. Thank you.
Cheers, Steve. There's
That was
also
Steve,
some of this.
right?
Hold position.
Just happy there's some action. I was about to fall asleep.
So yes, that's exactly what can happen. Just let us know either quick zoom or take a sample, and we can go from there.
Okay, is this a slurp?
This is a slurp, I think. It's been really effective lately.
Yeah, I recall one that we collected with the slurp during our last dive, Abby, I believe.
Yes.
So
Do
tell
we
our viewers a
want the
little bit about
lasers on
get,
and a zoom
yes,
before?
Yes,
Yeah,
definitely.
Yeah.
Thank you.
Yeah.
Zoom in now.
Abby, do you want the lasers on during the Zoom or off?
That's fine. On is good, yeah.
Once we zoom all the way in, we'll probably lose lasers anyways. So.
Yeah.
They're up there at the very top, tippy top.
More zoom, but
Right now, anyway.
I did. Wow.
Okay,
Look at
good
that. So
capture,
what we're looking
close up. There you go, lasers. Shot.
Laser zoom coming up. Here we go.
All right, laser
so I
shot
think
check.
you can even
I'm gonna pull out wide
see
body.
some little crustaceans in there.
Uh-huh.
So we're looking at a carnivorous sponge. So unlike other sponges, they don't filter feed. They were first discovered in Mediterranean caves in the 90s and tend to be found in sort of nutrient poor environments. So I think that this carnivorous lifestyle where they can capture prey is an adaptation to nutrient poor environments. And these particular types that look like little pipe cleaners with their spicules coming out, there's two genus that look like this, Asbestopluma and Cledorhiza.
All right, ready for section? Or
Yeah.
no?
Ready?
So the sample will help tell us which genus and which species we have at this particular area. You get branching ones and unbranched ones as well. So you get a little unbranched specimen.
And Abby, did the prey sort of impale themselves on the
Yep, there it
sponge or get
is.
stuck in those
Jar one.
spicules? Perfect.
And that is jar number 1.
Okay.
And if possible, before we move on, I can see that there's one of those button-like Thanea coral sponges on the rock just to the right. I don't know, Bob, if you think that could be if you might be able to take that sample as well. I was kind of hoping we might see one on a smaller cobble where the whole cobble could be picked up. It's definitely a tricky one to sample.
So not a slurp, it's a Pick up? With a rock?
I think it's a pickup. But I think that rock looks pretty large, so...
Oh, that's a huge rock.
Yeah. I don't know if... I don't know if you think you could scrape it off the rock or whether it's worth moving on and trying to find this one that's attached to a smaller piece of cobble that you think might be a good pickup.
Yeah. That's probably better. I don't know. I don't know how squishy it is.
From past experience, it's kind of brittle and crunchy, and it's a difficult one to bring up in one piece.
Okay. Yeah.
So
continue on.
All right, zooming out.
Yep.
Ready.
Steve, what was the sample number for the Abesta Pluma?
036.
Roger, thank you. Alrighty, moving on. Bridge nav.
Roger that. I'm typing, so I'm muted now.
Step 30 meters, bearing 325.
So Lisa's just reminding us to make sure we get zooms on the corals that we're coming across here.
Zoom in out.
Oh, what's that? Dead stock.
Ooh.
No good. Zoom out.
Looks like it might have been a Swifty in a former life.
Move underway.
Oh, yeah, it's a goner. But still providing habitat.
Need to reset when we're stopped again. We're moving. 325.
Another mushroom coral, Anthomastus. Is it still Anthomastus? I think.
Are we in transect now?
I remember when we did
There's
dive.
one of the things.
Is that on a smaller rock right there? I'll stop.
Nice. Take your pick, Abby.
Hold position.
My feeling is it's a bit jittery.
So it sounds
Zoom
jittery
in.
to you? Abby? Was that done? It was Abby.
You get a little everything on that one.
Yay.
Do you know what? I think it might be better to try and find one without so many different types on.
Too much.
What is this, the Goldilocks story?
Yeah.
Maybe. Well,
That's on a perfect rock.
is it?
It's okay, we can find another one.
Hang on.
But
Zoom out.
I did see just to the right that the white sponge that we've been seeing growing in plates. If you just look, maybe it's a bit
I gotta
up
move.
a little.
I gotta move to see it.
Okay.
Yeah, off to the right, the larger.
Yeah, that we had a nice zoom on
Plate.
before.
So if we're not on transect, I'll put up a telestrator.
Yeah, I think that's okay.
We're talking about right in
Here we
front of
go.
us right there?
So,
Or
so a
big-
little bit further up.
Okay.
So the one that looks like a fan that's kind of covered.
Portholes or something.
Yeah, that's the one. So I think that's something that we've been calling white Mycale in our AUV imagery, but we actually do not know what this is.
Now we have a
You've been
microscope.
waiting for that.
Now we have a microscope on the telestrator.
Oh,
That is
Better for
impressive.
worse.
Yeah, we had 3 hours in the midwater this morning, so I've
Zoom
got
in.
new toys on the telestrator.
Wow.
Ooh, that's really neat.
Yeah. So sponges have these tiny, tiny little pores on the outer surface called ostia, and water gets sucked in through there, pumped through the sponge's body by these wafting flagella, and then comes out the other side through these bigger holes called oscula.
You have stick lock on now, don't you? Holding us down.
So the big holes are the exits?
Big holes are the exit. It's kind of a one-way system. In through the tiny holes, the sponge gets its oxygen and its nutrients, and then pumps out the wastewater through these bigger oscula.
Wow. I should
It's a lovely
mention
shot.
it's the temperature here, which if you're watching, those of you watching from Nautilus Live, there are 4 channels you can look at, and actually the website itself will tell you the temperature, I think, and the depth in metric and English. So it'll give the depth in meters and feet, and the temperature in degrees Fahrenheit and degrees centigrade. It's 4.5 degrees centigrade is what we're seeing. And the oxygen saturation here is 1.3%. So this is a pretty, all these deep dives we've been doing have pretty low oxygen environment. It speaks to the adaptations these animals have. And just to remind, corals and sponges are colonial animals, correct, Abby? You want to talk a little bit more about that? What a sponge is, a colony of...
Yeah.
So are we getting a sample here? Or
I would love to get a sample here. Yes.
Okay.
Yeah. When you...
Thank you.
Sorry.
And I think it will be...
Lens laser
I think it'll be
shot first?
Yes, please. We've had some lovely
good
zooms.
lasers are on, yeah.
Yeah. And you
You
got
want
it?
them without?
Ready?
One without?
Right here. Here we go.
Without?
Nice.
All set.
Nice.
Right.
Okay.
Thank you.
So is this a grab or a,
I think this is a grab.
like the whole thing, or trying for the rock?
If you can get the rock, that's always a great way to do it. Otherwise, I'm thinking maybe that piece on the left could maybe try and grab, but
Who's typing?
I
Abby, are you typing?
nope.
Oh, it's me. I'll mute. Sorry.
But that's odd, though. It's the type of audio I wouldn't have expected.
Okay, zoom in.
Zoom in.
Oh, wait, I gotta get planted here.
And thank you, Di.
I took off the... We were shaking around, so I took off the power.
I saw that. Thank you.
So Guy Cochran is
Yeah,
saying,
you can
if
zoom
we
in.
are in Talus here, we need to go upslope to find bedrock when we're ready.
That sounds good. So, Di, to answer your question, so sponges so corals are colonial animals that when you look up close, you can see all the little polyps, and each one is a little individual animal with little stinging tentacles that it uses to capture prey. So sponges are not actually colonial like that. Sort of each sponge is an individual made up of different cells. So they're not colonial in the same way that corals are.
Yeah, so that is a loose rock. I think I can get the whole thing. Is that what we're going for, the rock?
That sounds good.
All right.
I think this is the advantage of being on the first shift, as you can fill up the boxes.
So it looks like there's 2 pieces separated. So those would be 2 individual sponges, Abby? Or maybe they broke away.
Yeah, they're kind of attached to the side rocks, so
yeah.
in
So when you're doing sponge surveys, that is something that different researchers and different research groups will approach differently. I know when I was doing sponge counts, I was counting individual patches, but you can also just sort of use them, like calculate surface area covered
Go
by
to starboard side.
Starboard?
Yeah.
There's other situations. It's got two bonus, the either tunicates or encrusting sponges as well. Yeah.
The two little yellow button ones underneath.
Yeah, I think we can go in E or F on the starboard side for this one.
Okay.
So, yeah, in some situations, it might be almost impossible to tell where one sponge starts and the next one ends. It can also be reproduced by
now.
pudding.
Gotta change the cameras too. Oh no, they're good.
Do we have an ID for this, or at least
we'll have to open them,
family maybe?
Boxes.
Well, I would say it's a demo. Okay, I'm putting in the chat now how I'm going to record it.
Ready? Or let me know when you're ready.
Get more organized here. Okay, box out. You want an F?
We have the scoop and F.
Yeah, I got the scoop and F.
All right, E then.
Hercules, the ROV, is equipped with a bunch of things, including bio boxes, we call them, and they're talking about, the pilots are talking about which portion, which section of the box that sample is going to go into.
Alrighty, shoreside. So do we want to continue up towards the next waypoint 3C? Currently we're on a slight northwest heading to follow this contour line up to 3C.
Are we on a little bit off the ridge, off the side of the ridge? Guy Cochran is saying if we're in Talus here, we need to go upslope to find bedrock.
Sure, we can pop up to one of the contours that's directly north of Hercules right now, so we can continue kind of straight from where we did the transect up to one of these higher contours, and then move across northwest to 3C.
Yeah, let's see if we can find some bedrock up at the top, and then we'll proceed toward 3C.
Roger.
Yeah, he's saying yes, straight north, not northeast yet.
Okay, straight north. Bridge now.
Thank you, Guy.
Uh, 30 meters, bearing 053.
Thank you, Stan.
Actually, I just realized that I was saying something different to you in the bridge, so stand by on that one. Bridge nav.
Are we still going after the little round sponge?
30 meters, bearing 0.
Yeah, right there near the lasers.
Like maybe this, yeah.
Sorry.
All right.
Ooh.
I know. Oh, bridge nav.
Oh dear. Okay.
Hold position. Well, that gives me a little more time to figure out what we want to do here.
So what are we doing? I think that's on a
Are we getting that little yellow
rock. I'm going to zoom in now.
Yeah.
More dirt.
Excellent.
We're okay with that one?
Yeah, that looks ideal.
Okay. Hopefully that's a loose rock, not doesn't have a big root. Yeah.
And are these more the little yellow sponges? What are those to the left?
Those are bonus sponges.
Bonus sponges, I like it.
I don't know if those, yeah, we've seen those yellow things on other rocks. I'm not sure what they are. I think there might be tunicates or eggs.
Tuna.
Where do we think this one's gonna go?
Can you zoom out now?
Yeah, I mean, I guess we're gonna have to go in the forward box.
Is the porch light on?
Yeah, it's on.
Okay, thank you. We've got to do something.
Okay, zoom in.
One of the ways I used to check if something was a sponge or a tunicate, if I wasn't sure, was to poke the give it a scientific poke, and the tunicates will often retract.
Do we need that? A scientific poke?
You could poke the little yellow thing. Well, I think it does look like eggs, perhaps.
Wow.
Yeah.
Just... you broke it.
It didn't seem to complain very much.
No.
What's that thing over to the left, right under the claw now? A little bit left. Strange looking. Looks like a doily on a rock.
Oh, brilliant.
Ooh, well done.
Not in a
Still holding my
box.
breath.
You had good grabs?
Yep. All set there.
All right, zooming out.
Yeah, we can go in either of the forward compartments.
Okay.
Are you ready for the box out?
Yep. Little yellow dude got away in there.
There's one.
I'm trying to look up who we see off there to the right.
Which are you looking at, Di?
Oh, I'm not right now. The white coral.
Hey,
Fantastic
right
sample.
now. Okay. Yay, well done.
Yes, thank you.
That specimen straight ahead. I don't know who that is. I'm trying to look it up.
Zoom in now.
Sure. If you're asking.
Pan down to the base.
Yeah. Is it a bamboo?
Yeah, that's a bamboo. It's a Isidella.
Yeah. Yeah.
All right, good capture.
Okay. Zoom out.
What do you think? What do you think, Di?
Well, I'm thinking we should collect a piece. We're not getting any feedback from the chat.
Yeah.
I haven't looked at the list. I was just trying to look up what it was. But I think Meredith.
There are. Yes, all of the bamboos on this list.
Okay, now I'm on the list. It looks like we should get it. Is that what you're
Yep.
thinking?
Mm-hmm.
A piece?
I gotta
Okay.
set up a little different here.
Another sample.
Okay.
Ship stopped. We can stay as long as you want.
Thank you.
So with these, I just recommend to get at least
Slurp.
1 or 2 branch points.
Okay.
So it's going to be a cut.
Yep.
Thank you, Steve.
Yeah.
Steve, is this a small enough move that we can keep the same target?
I think so.
Great.
After all, it doesn't really mean anything, right?
Hey. You're not wrong.
It's just our perception of 3D space on a 2D screen.
Okay, zoom in now.
It's all a construct, man.
I was going to say, yeah, we're all an illusion. Isidella, right?
Yeah.
Stella, okay.
And is this going in a box?
Yeah,
We
I
don't
think
have a
it'll
great
go in a
picture that
box.
looks like this in our list, do we, Abby?
No, but so the ones on that list are the ones that are permitted within the sanctuary. So I do,
Yeah.
I know that when I was asking Meredith, there are a lot more species of interest kind of outside the sanctuary as well.
Yeah.
I mean, now that we're outside, that we don't necessarily need to be on the permit.
Ah, so that's why we're sampling so much.
Is that gonna be a good piece in there?
I think that'll be okay.
Do you want to tilt down? Ben, can
You
Paul,
pan me
reach
down
over?
just a skosh.
Yeah.
Perfect.
They're good right there.
Yep.
Yeah.
Where's this
Can we
going?
go starboard side in one of the small boxes?
Yeah. Okay. Is it floaty?
It's not a floaty, it'll sink.
Okay.
All right. Going over to sample.
So Steve, what kind of things do you look at when you're identifying these corals?
With this
Ready?
particular bamboo, it's one that we see quite frequently. So
Boxshell?
Isidella is has this kind of candelabra-like branching pattern.
Okay.
And the tentacles at the bottom are give it away also that it's one of a couple species.
Mm.
I mean, if I didn't know what it was, I would look closer at the node structure, the branching structure, whether it's branching at the node or the internode.
Okay.
You know, that separates a couple different categories of bamboo coral.
Mm. Mm-hmm.
And if
Oh, you lied.
what?
It's floaty. It's totally floaty.
Not usually floaty.
Oh my God, it's the one that got away.
No.
There's
Got
always
away.
one.
Well, I mean, it's not a rock, but
Uh-oh.
Oh, is that the current maybe in this?
Yeah, the current's blowing pretty good.
Yeah.
So shall we try to take another piece? Did we lose it entirely? I can't see.
It went inside the frame of the vehicle, blew inside.
Oh.
Yeah, it was in the box and then it
Floated away.
floated
Wasn't.
away.
One that got away.
Okay.
not the coral's fault.
No.
It's the water's fault.
What?
The
Not the Robert
not
waters.
the Bob waters, the sea
What about
water.
the scientists?
No, the
Not my
waters of
fault,
the
not the
ocean's
waters.
fault. Oh man. All you last name
Okay,
people
so
with waters last name.
Are we giving it another haircut here?
Yeah, might as well. We're here, ready to go.
Think
All right.
so.
yep.
Okay, zoom in now. Well, other side, perhaps? It's
Sure.
gonna be
That seems
harder to see, maybe.
So I've got my brother texting me asking why we call them bamboo corals. And I think it's because when the coral is dead, you can see the skeleton kind of looks like bamboo, like
Can you
white
pan
picky
me up
little
just a
brown
skosh?
bands.
Right.
Oh,
And that when we were looking
wrong way.
down toward the base, I could see one of those nodes more easily, and that's what said bamboo to me, my lesser trained eye.
Yeah, the last character I
Maybe
was
a tilt down's
mentioning
in order
is
now,
that
Paul.
you know, branching pattern, branching structure is sometimes unreliable. So the actual shape of the polyp and the sclerites that may be protruding from the polyp are
Can you give me a little
somewhat
more down?
reliable
Yep.
Can't tell if
characters for determining different
I don't wanna
cleats.
grab the rest of it here.
All right, why don't we go with that into the sponge compartment that we just put in the forward box.
Okay, zoom out. Okay, box out.
Yep.
Go, 18. Don't you float.
Mm-mm.
Caught in my teeth.
It's like, yeah, can't shake it off.
Oh no.
Uh-oh.
It'll be all
Got a
right.
Can you
toothpick?
scrape it on the
going.
top of the box there?
Oh,
We'll get it out.
the good old shake it. Nice.
Oof.
Well done.
Oh, good.
Is that some sort of crustacean eating a fish right to our left
Yeah,
right now?
we killed we accidentally.
Oh, it sure looks like it.
Yeah.
Shall we zoom
Yeah,
in
he
and
got
have
a
a look
bonus.
before we?
You got a ROV supplied bonus.
Al,
Is-
can we get a zoom?
What happens when we're going through those big clouds of
What am I zooming in on? Oh, that thing.
You'll see.
a little meal?
He's just
We
hugging
got
him.
an
Yeah, it's a hug.
Just hugging it.
Totally a hug.
The poor fish.
This is a Halloween hug.
That fish looks thrilled.
It's caressing him too. Oh, this is sick.
Okay.
Huh.
I regret.
This is
Mentioning
the
it.
stuff that goes viral. You're gonna want this.
That's an anchovy.
Or it was.
Was an anchovy.
Oh.
Yeah.
Oh,
Yeah.
Oh.
Oh no.
Oh, I think it's deceased. It's the departed now.
this isn't going to end well. All right, science, are we ready to get going?
Okay. Yes.
Oh, the shrimp on the other end is going for it too.
Yep.
Yeah.
Okay, circle of life.
The circle of life, the food web.
Are we ready?
Okay, so we're headed upslope to hopefully bedrock.
Okay, so we're going to go directly north from where we currently are. Let's see if we can get past 20 meters from where we originally ended our transect.
Yes. Should we cover our eyes?
Do you want to?
Yeah.
Do you want to look one more time?
Oh, anything else?
Okay, no, we better get going.
Okay, bridge, Nav.
I will.
Step 30 meters, bearing 325. Alrighty, ship move
Okay.
is being put in right now, and we've got a pilot switch, so Paul's on Hercules now.
Hi, Paul.
Hi.
Bye, Bob. Bye, Bob.
He's still here.
Oh, Bob's still
Watch
there.
what you say about him. Oh,
About
we
to
had
correct
decided to go.
myself. I was. Oh, go ahead.
Sorry, bridge. How about bearing of 0? Wrote down the wrong number.
All right.
Sure it is.
Due north.
I think you got to go that way.